And if you watch patiently, you may see a cell slowly change shape and divide into two. See Figure 1.4 and the speeded up video of cell division in a frog embryo in Movie 1.1. To see the internal structure of a cell is difficult, not only because the parts are small, but also because they are transparent and mostly colorless. One approach is to stain cells with dyes that color particular components differently. See Figure 1.5. Alternatively, one can exploit the fact that cell components differ slightly from one another in refractive index, just as glass differs in refractive index from water, causing light rays to be deflected as they pass from the one medium into the other. The small differences in refractive index can be made visible by specialized optical techniques, and the resulting images can be enhanced further by electronic processing. See Panel 1.1, pages 8 and 9. The cell thus revealed has a distinct anatomy, Figure 1.7. It has a sharply defined boundary, indicating the presence of an enclosing membrane. A large round body, the nucleus, is prominent in the middle of the cell. Around the nucleus and filling the cell's interior lies the cytoplasm, a transparent substance crammed with what seems at first to be a jungle of miscellaneous objects. With a good light microscope, one can begin to distinguish and classify specific components in the cytoplasm. Figure 1.7b. However, structures smaller than about 0.2 micrometers, about half of the wavelength of visible light, cannot be resolved with a conventional light microscope. Points closer than this are not distinguishable and appear as a single blur. In the past few years, new types of fluorescence microscopes have been developed. Using sophisticated methods of illumination and image analysis to see detail several times finer than this. However, for the highest magnification and the best resolution, one must turn to an electron microscope, which can reveal details down to a few nanometers, or nm. See Figure 1.6. Cell samples for the electron microscope require painstaking preparation. Even for light microscopy, a tissue usually has to be fixed, that is, preserved by pickling in a reactive chemical solution, supported by embedding in a solid wax or resin, cut or sectioned into thin slices, and stained before it is viewed. For electron microscopy, similar procedures are required, but the sections have to be much thinner and there is no possibility of looking at living wet cells. When thin sections are cut, stained, and placed in the electron microscope, much of the jumble of cell components becomes sharply resolved into distinct organelles, separate, recognizable substructures that are only hazily defined under the light microscope. A delicate membrane about 5 nanometers thick is visible enclosing the cell, and similar membranes form the boundary of many of the organelles inside. Figure 1.8ab. The external membrane is called the plasma membrane, while the membranes surrounding organelles are called internal membranes. With an electron microscope, even some of the large molecules in a cell can be seen individually. Figure 1.8c. The type of electron microscope used to look at thin sections of tissue is known as a transmission electron microscope. This is, in principle, similar to a light microscope, except that it transmits a beam of electrons rather than a beam of light through the sample. Another type of electron microscope, the scanning electron microscope, scatters electrons off the surface of the sample and so is used to look at the surface detail of cells and other structures. See Panel 1.1, pages 8 to 9. Electron microscopy enables biologists to see the structure of biological membranes, which are only 2 molecules thick, discussed in detail in Chapter 11. Even the most powerful electron microscopes, however, cannot reveal the individual atoms that make up molecules. Figure 1.9. The microscope is not the only tool that modern biologists use to study the details of cell components. Techniques such as X-ray crystallography, for example, can be used to determine the three-dimensional structure of protein molecules, discussed in Chapter 4. We shall describe other methods for probing the inner workings of cells as they arise throughout the book. The prokaryotic cell. Of all the types of cells revealed by the microscope, bacteria have the simplest structure and come closest to showing us life stripped down to its essentials. Indeed, a bacterium contains essentially no organelles, not even a nucleus to hold its DNA. This property, the presence or absence of a nucleus, is used as the basis for a simple but fundamental classification of all living things. Organisms whose cells have a nucleus are called eukaryotes, from the Greek word eu meaning well or truly, and karyon, a kernel or nucleus. Organisms whose cells do not have a nucleus are called prokaryotes, from pro meaning before. The terms bacterium and prokaryote are often used interchangeably, although we shall see that the category of prokaryotes also includes another class of cells, the archaea, single archaeon, which are so remotely related to bacteria that they are given a separate name. Prokaryotes are typically spherical, rod-like, or corkscrew-shaped, and small, just a few micrometers long. Figure 110. Although there are some giant species as much as 100 times longer than this. They often have a tough protective coat, called a cell wall, surrounding the plasma membrane, which encloses a single compartment containing the cytoplasm and the DNA. In the electron microscope, the cell interior typically appears as a matrix of varying texture without any obvious organized internal structure. Figure 111. The cells reproduce quickly by dividing in two. Under optimum conditions, when food is plentiful, a prokaryotic cell can duplicate itself in as little as 20 minutes. In 11 hours, by repeated divisions, a single prokaryote can give rise to more than 8 billion progeny, which exceeds the total number of humans presently on Earth. Thanks to their large numbers, rapid growth rates, and ability to exchange bits of genetic material by a process akin to sex, populations of prokaryotic cells can evolve fast, rapidly acquiring the ability to use a new food source or to resist being killed by a new antibiotic. Prokaryotes are the most diverse of cells. Most prokaryotes live as single-celled organisms, although some join together to form chains, clusters, or other organized multicellular structures. In shape and structure, prokaryotes may seem simple and limited, but in terms of chemistry, they are the most diverse and inventive class of cells. These creatures exploit an enormous range of habitats, from hot puddles of volcanic mud to the interiors of other living cells, and they vastly outnumber other living organisms on Earth. Some are aerobic, using oxygen to oxidize food molecules. Some are strictly anaerobic, and are killed by the slightest exposure to oxygen. As we discuss later in this chapter, mitochondria, the organelles that generate energy for the eukaryotic cell, are thought to have evolved from aerobic bacteria that took to living inside the anaerobic ancestors of today's eukaryotic cells. Thus, our own oxygen-based metabolism can be regarded as a product of the activities of bacterial cells. Virtually any organic material, from wood to petroleum, can be used as food by one sort of bacterium or another. Even more remarkably, some prokaryotes can live entirely on inorganic substances. They can get their carbon from CO2 in the atmosphere, their nitrogen from atmospheric N2, and their oxygen, hydrogen, sulfur, and phosphorus from air, water, and inorganic materials. Some of these prokaryotic cells, like plant cells, perform photosynthesis, getting energy from sunlight. Figure 112. Others derive energy from the chemical reactivity of inorganic substances in the environment. Figure 113. In either case, such prokaryotes play a unique and fundamental part in the economy of life on Earth. Other living things depend on the organic compounds that these cells generate from inorganic materials. Plants, too, can capture energy from sunlight and carbon from atmospheric CO2. But plants unaided by bacteria cannot capture N2 from the atmosphere, and in a sense, even plants depend on bacteria for photosynthesis. It is almost certain that the organelles in the plant cell that perform photosynthesis, the chloroplasts, have evolved from photosynthetic bacteria that found a home inside the plant cell's cytoplasm. The world of prokaryotes is divided into two domains, bacteria and archaea. Traditionally, all prokaryotes have been classified together in one large group, but molecular studies reveal that there is a gulf within the class of prokaryotes, dividing it into two distinct domains called the bacteria, or sometimes eubacteria, and the archaea. Remarkably, at a molecular level, the members of these two domains differ as much from one another as either does from the eukaryotes. Most of the prokaryotes familiar from everyday life, the species that live in the soil or make us ill, are bacteria. Archaea are found not only in these habitats, but also in environments hostile to most other cells. There are species that live in concentrated brine, in hot, acidic volcanic springs, in the airless depths of marine sediments, in the sludge of sewage treatment plants, in pools beneath the frozen surface of Antarctica, and in the acidic, oxygen-free environment of a cow's stomach, where they break down cellulose and generate methane gas. Many of these environments resemble the harsh conditions that must have existed on the primitive Earth, where living things first evolved, before the atmosphere became rich in oxygen. The eukaryotic cell. Eukaryotic cells in general are bigger and more elaborate than bacteria and archaea. Some live independent lives as single-celled organisms, such as amoebae and yeasts. Figure 114. Others live in multicellular assemblies. All of the more complex multicellular organisms, including plants, animals, and fungi, are formed from eukaryotic cells. By definition, all eukaryotic cells have a nucleus, but possession of a nucleus goes hand in hand with possession of a variety of other organelles, subcellular structures that perform specialized functions. Most of these are likewise common to all eukaryotic organisms. We will now take a look at the main organelles found in eukaryotic cells from the point of view of their functions. The nucleus is the information store of the cell. The nucleus is usually the most prominent organelle in an eukaryotic cell. Figure 115. It is enclosed within two concentric membranes that form the nuclear envelope, and it contains molecules of DNA, extremely long polymers that encode the genetic information of the organism. In the light microscope, these giant DNA molecules become visible as individual chromosomes when they become more compact as a cell prepares to divide into two daughter cells. Figure 116. DNA also stores the genetic information in prokaryotic cells. These cells lack a distinct nucleus, not because they lack DNA, but because they do not keep their DNA inside a nuclear envelope, segregated from the rest of the cell contents. Mitochondria generate usable energy from food to power the cell. Mitochondria are present in essentially all eukaryotic cells, and they are among the most conspicuous organelles in the cytoplasm. Figure 117. These organelles have a very distinctive structure when seen with an electron microscope. Each mitochondrion appears sausage or worm-shaped. from 1 to many micrometers long, and each is enclosed in two separate membranes. The inner membrane is formed into folds that project into the interior of the mitochondrion. Figure 118. Mitochondria contain their own DNA and reproduce by dividing in two. Because mitochondria resemble bacteria in so many ways, they are thought to derive from bacteria that were engulfed by some ancestor of present-day eukaryotic cells. Figure 119. This evidently created a symbiotic relationship in which the host eukaryote and the engulfed bacterium helped one another to survive and reproduce. Observation under the microscope by itself gives little indication of what mitochondria do. Their function was discovered by breaking open cells and then spinning the soup of cell fragments in a centrifuge. This separates the organelles according to their size, shape, and density. Purified mitochondria were then tested to see what chemical processes they could perform. This revealed that mitochondria are generators of chemical energy for the cell. They harness the energy from the oxidation of food molecules, such as sugars, to produce adenosine triphosphate, or ATP, the basic chemical fuel that powers most of the cell's activities. Because the mitochondrion consumes oxygen and releases carbon dioxide in the course of this activity, the entire process is called cellular respiration, essentially breathing on a cellular level. The process of cellular respiration is considered in detail in Chapter 14. Without mitochondria, animals, fungi, and plants would be unable to use oxygen to extract the maximum amount of energy from the food molecules that nourish them. Oxygen would be a poison for them, rather than an essential requirement. That is, they would be anaerobic. Many prokaryotes are anaerobic, and there are even a few anaerobic eukaryotes, such as the intestinal parasite Giardia, that lack mitochondria and live only in environments that are low in oxygen. Chloroplasts capture energy from sunlight. Chloroplasts are large green organelles that are found only in the cells of plants and algae, not in the cells of animals or fungi. These organelles have an even more complex structure than mitochondria. In addition to their two surrounding membranes, chloroplasts possess internal sacs of membranes containing the green pigment chlorophyll. When a plant is kept in the dark, its greenness fades. When put back in the light, its greenness returns. This suggests that the chlorophyll and the chloroplasts that contain it are crucial to the special relationship that plants and algae have with light. But what is that relationship? Animals and plants all need energy to live, grow, and reproduce. Animals can use only the chemical energy they obtain by feeding on the products of other living things. But plants can get their energy directly from sunlight, and chloroplasts are the organelles that enable them to do so. From the standpoint of life on Earth, chloroplasts carry out an even more essential task than mitochondria. They perform photosynthesis, that is, they trap the energy of sunlight in chlorophyll molecules and use this energy to drive the manufacture of energy-rich sugar molecules. In the process, they release oxygen as a molecular byproduct. Plant cells can then extract the stored chemical energy when they need it, by oxidizing these sugars in their mitochondria, just as animal cells do. Chloroplasts thus generate both the food molecules and the oxygen that all mitochondria use. How they do so is explained in Chapter 14. Like mitochondria, chloroplasts contain their own DNA, reproduced by dividing in two, and are thought to have evolved from bacteria. In this case, from photosynthetic bacteria that were somehow engulfed by an early eukaryotic cell. Figure 121. Internal membranes create intracellular compartments with different functions. Nuclei, mitochondria, and chloroplasts are not the only membrane-enclosed organelles inside eukaryotic cells. The cytoplasm contains a profusion of other organelles, most of them surrounded by single membranes, that perform many distinct functions. Most of these structures are involved with the cell's ability to import raw materials and to export manufactured substances and waste products. Some of these membrane-enclosed organelles are enormously enlarged in cells that are specialized for the secretion of proteins. Others are particularly plentiful in cells specialized for the digestion of foreign bodies. The endoplasmic reticulum, ER, an irregular maze of interconnected spaces enclosed by a membrane, figure 122, is the site where most cell membrane components, as well as materials destined for export from the cell, are made. Stacks of flattened membrane-enclosed sacs constitute the Golgi apparatus, figure 123, which receives and often chemically modifies the molecules made in the endoplasmic reticulum, and then directs them to the exterior of the cell or to various locations inside the cell. Lysosomes are small, irregularly shaped organelles in which intracellular digestion occurs, releasing nutrients from food particles and breaking down unwanted molecules for recycling or excretion. And peroxisomes are small, membrane-enclosed vesicles that provide a contained environment for reactions in which hydrogen peroxide, a dangerously reactive chemical, is generated and degraded. Membranes also form many different types of small vesicles involved in the transport of materials between one membrane-enclosed organelle and another. This whole system of related organelles is sketched in figure 124a. A continual exchange of materials takes place between the endoplasmic reticulum, the Golgi apparatus, the lysosomes, and the outside of the cell. The exchange is mediated by small vesicles that pinch off from the membrane of one organelle and fuse with another, like tiny soap bubbles budding from and rejoining larger bubbles. At the surface of the cell, for example, portions of the plasma membrane tuck inward and pinch off to form vesicles that carry material captured from the external medium into the cell. Figure 125. These vesicles fuse with membrane-enclosed endosomes, which mature into lysosomes, where the imported material is digested. Animal cells can engulf very large particles, or even entire foreign cells, by this process of endocytosis. The reverse process, exocytosis, whereby vesicles from inside the cell fuse with the plasma membrane and release their contents into the external medium, is also a common cellular activity. See figure 125. Most hormones, neurotransmitters, and other signaling molecules are secreted from cells by exocytosis. How the membrane-enclosed organelles transport proteins and other molecules from place to place inside the cell is discussed in more detail in Chapter 15. The cytosol is a concentrated aqueous gel of large and small molecules. If we were to strip the plasma membrane from a eukaryotic cell and then remove all of its membrane-enclosed organelles, including nucleus, endoplasmic reticulum, Golgi apparatus, mitochondria, chloroplasts, and so on, we would be left with the cytosol. See figure 124b. In other words, the cytosol is the part of the cytoplasm that is not partitioned off within intracellular membranes. In most cells, the cytosol is the largest single compartment. It contains a host of large and small molecules crowded together so closely that it behaves more like a water-based gel than a liquid solution. Figure 126. The cytosol is the site of many chemical reactions that are fundamental to the cell's existence. The early steps in the breakdown of nutrient molecules take place in the cytosol, for example, and it is here that the cell performs one of its key synthetic processes, the manufacture of proteins. Ribosomes, the molecular machines that make the protein molecules, are visible with the electron microscope as small particles in the cytosol, often attached to the cytosolic phase of the endoplasmic reticulum. See Figure 18b and 122b. The cytoskeleton is responsible for directed cell movements. The cytoplasm is not just a structureless soup of chemicals and organelles. Using an electron microscope, one can see that in eukaryotic cells, the cytosol is crisscrossed by long, fine filaments of protein. Frequently, the filaments are seen to be anchored at one end of the plasma membrane or to radiate out from a central site adjacent to the nucleus. This system of filaments is called the cytoskeleton, Figure 127. The thinnest of the filaments are actin filaments, which are present in all eukaryotic cells, but occur in especially large numbers inside muscle cells, where they serve as part of the machinery that generates contractile forces. The thickest filaments are called microtubules because they have the form of minute hollow tubes. In dividing cells, they become reorganized into a spectacular array that helps pull the duplicated chromosomes in opposite directions and distribute them equally to the two daughter cells. Figure 128. Intermediate in thickness between actin filaments and microtubules are the intermediate filaments, which serve to strengthen the cell mechanically. These three types of filaments, together with other proteins that attach to them, form a system of girders, ropes, and motors that gives the cell its mechanical strength, controls its shape, and drives and guides its movements. See Movie 1.2 and Movie 1.3. Because the cytoskeleton governs the internal organization of the cell as well as its external features, it is as necessary to a plant cell boxed in by a tough wall of extracellular matrix as it is to an animal cell that freely bends, stretches, swims, or crawls. In a plant cell, for example, organelles such as mitochondria are driven in a constant stream around the cell interior along cytoskeletal tracts, and animal cells and plant cells alike depend on the cytoskeleton to separate their internal components into two daughter sets during cell division. Its role in cell division may be the most ancient function of the cytoskeleton. Even bacteria contain proteins that are distantly related to those of eukaryotic actin filaments and microtubules and that form filaments that play a part in prokaryotic cell division. We examine the cytoskeleton in detail in Chapter 17 and discuss its role in cell division in Chapter 18 and its responses to signals from the environment in Chapter 16. The cytoplasm is far from static. The cell interior is in constant motion. The cytoskeleton is a dynamic jungle of ropes and rods that are continually being strung together and taken apart. Its filaments can assemble and then disappear in a matter of minutes. Along these tracks and cables, organelles and vesicles hurry to and fro, racing across the width of the cell in a second or so. The endoplasmic reticulum and the molecules that fill every free space are in frantic thermal commotion, with unattached proteins buzzing around so fast that even though they move at random, they visit every corner of the cell within a few seconds, constantly colliding with an even more tumultuous dust storm of smaller organic molecules. Of course, neither the bustling nature of the cell's interior nor the details of cell structure were appreciated when scientists first peered at cells in a microscope. Our knowledge of cell structure accumulated slowly. A few of the key discoveries are listed in Table 1.1. Panel 1.2 summarizes the differences between animal, plant, and bacterial cells. Eukaryotic cells may have originated as predators. Eukaryotic cells are typically 10 times the length and 1,000 times the volume of prokaryotic cells. although there is huge size variation within each category. In addition, they possess a whole collection of features, a cytoskeleton, mitochondria, and other organelles, that set them apart from bacteria and archaea. When and how eukaryotes evolved these systems remains something of a mystery. Although eukaryotes, bacteria, and archaea must have diverged from one another very early in the history of life on Earth, discussed in Chapter 14, the eukaryotes did not acquire all of their distinctive features at the same time. Figure 129. According to one theory, the ancestral eukaryotic cell was a predator that fed by capturing other cells. Such a way of life requires a large size, a flexible membrane, and a cytoskeleton to help the cell move and eat. The nuclear compartment may have evolved to keep the DNA segregated from this physical and chemical hurly-burly, so as to allow more delicate and complex control of the way the cell reads out its genetic information. Such a primitive eukaryote with a nucleus and cytoskeleton was most likely the sort of cell that engulfed the free-living, oxygen-consuming bacteria that were the ancestors of the mitochondria. See Figure 119. This partnership is thought to have established 1.5 billion years ago, when the Earth's atmosphere first became rich in oxygen. A subset of these cells later acquired chloroplasts by engulfing photosynthetic bacteria. See Figures 121 and 129. That single-celled eukaryotes can prey upon and swallow other cells is borne out by the behavior of many of the free-living, actively motile microorganisms called protozoans. Didymium, for example, is a large carnivorous protozoan with a diameter of about 150 micrometers, roughly 10 times that of the average human cell. It has a globular body encircled by two fringes of cilia, and its front end is flattened except for a single protrusion rather like a snout. Figure 130. Didymium swims at high speed by means of its beating cilia. When it encounters a suitable prey, usually another type of protozoan, it releases numerous small paralyzing darts from its snout region. Didymium then attaches to and devours the other cell, inverting like a hollow ball to engulf its victim, which is almost as large as itself. Protozoans include some of the most complex cells known. Figure 131 conveys something of the variety of forms of protozoans, and their behavior is just as diverse. They can be photosynthetic or carnivorous, motile or sedentary. Their anatomy is often elaborate and includes such structures as sensory bristles, photoreceptors, beating cilia, stalk-like appendages, mouth parts, stinging darts, and muscle-like contractile bundles. Although they are single cells, protozoans can be as intricate and versatile as many multicellular organisms. Model organisms. All cells are thought to be descended from a common ancestor, whose fundamental properties have been conserved through evolution. Thus, knowledge gained from the study of one organism contributes to our understanding of others, including ourselves. But certain organisms are easier than others to study in the laboratory. Some reproduce rapidly and are convenient for genetic manipulations. Others are multicellular but transparent, so that one can directly watch the development of all their internal tissues and organs. For reasons such as these, large communities of biologists have become dedicated to studying different aspects of the biology of a few chosen species, pooling their knowledge to gain a deeper understanding than could be achieved if their efforts were spread over many different species. Information obtained from these studies contributes to our understanding of how all cells work. Although the roster of these representative organisms is continually expanding, a few stand out in terms of the breadth and depth of information that has been accumulated about them over the years. In the following sections, we examine some of these model organisms and review the benefits each offers to the study of cell biology, and in many cases, to the promotion of human health. Molecular biologists have focused on E. coli. In the world of bacteria, the spotlight of molecular biology has fallen chiefly on just one species, Escherichia coli, or E. coli for short. See Figure 111. This small, rod-shaped bacterial cell normally lives in the gut of humans and other vertebrates, but it can be grown easily in a simple nutrient broth in a culture bottle. E. coli copes well with variable chemical conditions in its environment, and it reproduces rapidly. Its genetic instructions are contained in a single, circular, double-stranded molecule of DNA, approximately 4.6 million nucleotide pairs long, and it makes 4,300 different kinds of proteins. In molecular terms, we understand the workings of E. coli more thoroughly than those of any other living organism. Most of our knowledge of the fundamental mechanisms of life, including how cells replicate their DNA and how they decode these genetic instructions to make proteins, has come from studies of E. coli. Subsequent research has confirmed that these basic processes occur in essentially the same way in our own cells as they do in E. coli. Brewer's yeast is a simple eukaryotic cell. We tend to be preoccupied with eukaryotes because we are eukaryotes ourselves. But human cells are complicated and difficult to work with, and if we want to understand the fundamentals of eukaryotic cell biology, it is often more effective to concentrate on a species that, like E. coli among the bacteria, is simple and robust and reproduces rapidly. The popular choice for this role of minimal model eukaryote has been the budding yeast, Saccharomyces cerevisiae, Figure 132, the same microorganism that is used for brewing beer and baking bread. S. cerevisiae is a small, single-celled fungus, and thus, according to modern views, is at least as closely related to animals as it is to plants. Like other fungi, it has a rigid cell wall, is relatively immobile, and possesses mitochondria, but not chloroplasts. When nutrients are plentiful, it reproduces almost as rapidly as a bacterium, as its nucleus contains only about 2.5 times as much DNA as E. coli. This yeast is also a good subject for genetic analysis. Even though its genome is small, by eukaryotic standards, S. cerevisiae carries out all the basic tasks every eukaryotic cell must perform. Genetic and biochemical studies in yeast have been crucial to understanding many basic mechanisms in eukaryotic cells, including the cell division cycle, the chain of events by which the nucleus and all the other components of a cell are duplicated and parceled out to create two daughter cells. In fact, the machinery that governs cell division has been so well conserved over the course of evolution that many of its components can function interchangeably in yeast and human cells. If a mutant yeast lacks a gene essential for cell division, providing it with a copy of the corresponding gene from a human will cure the yeast's defect and enable it to divide normally. See how we know. Pages 30 to 31. Arabidopsis has been chosen out of 300,000 species as a model plant. The large multicellular organisms that we see around us, both plants and animals, seem fantastically varied, but they are much closer to one another in their evolutionary origins and more similar in their basic cell biology than the great host of microscopic single-cell organisms. Whereas bacteria, archaea, and eukaryotes separated from each other more than 3 billion years ago. Plants, animals, and fungi are separated by only about 1.5 billion years. Fish and mammals by only about 450 million years, and the different species of flowering plants by less than 200 million years. The close evolutionary relationship among all flowering plants means that we can get insight into their cell and molecular biology by focusing on just a few convenient species for detailed analysis. Out of the several 100,000 species of flowering plants on Earth today, molecular biologists have recently focused their efforts on a small weed, the common wallcress, Arabidopsis thaliana, Figure 133, which can be grown indoors in large numbers and produces thousands of offspring per plant within 8 to 10 weeks. Arabidopsis has a genome of approximately 110 million nucleotide pairs, about 8 times as many as yeast, and its complete sequence is known. By examining the genetic instructions that Arabidopsis carries, we are beginning to learn more about the genetics, molecular biology, and evolution of flowering plants, which dominate nearly every ecosystem on land. Because genes found in Arabidopsis have counterparts in agricultural species, studying this simple weed provides insights into the development and physiology of the crop plants upon which our lives depend, as well as all the other plant species that are our companions on Earth. The world of animals is represented by a fly, a worm, a fish, a mouse, and the human species. Multicellular animals account for the majority of all named species of living organisms, and the majority of animal species are insects. It is fitting, therefore, that an insect, the small fruit fly Drosophila melanogaster, Figure 134, should occupy a central place in biological research. In fact, the foundations of classical genetics were built to a large extent on studies of this insect. More than 80 years ago, studies of the fruit fly provided definitive proof that genes, the units of heredity, are carried on chromosomes. In more recent times, a concentrated systematic effort has been made to elucidate the genetics of Drosophila, and especially how the genetic mechanisms underlying its embryonic and larval development. Through this work, we are at last beginning to understand in detail how living cells achieve their most spectacular feat, how a single fertilized egg cell, or zygote, develops into a multicellular organism, comprising vast numbers of cells of different types, organized in an exactly predictable way. Drosophila mutants with body parts strangely misplaced or oddly patterned have provided the key to identifying and characterizing the genes that are needed to make a properly structured adult body, with gut, wings, legs, eyes, and all the other bits and pieces in their correct places. These genes, which are copied and passed on to every cell in the body, define how each cell will behave in its social interactions with its sisters and cousins. In this way, they control the structures that the cells create. Drosophila, more than any other organism, has shown us how to trace the chain of cause and effect from the genetic instructions encoded in the DNA to the structure of the adult multicellular organism. Moreover, the genes of Drosophila have turned out to be amazingly similar to those of humans, far more similar than one would suspect from outward appearances. Thus, the fly serves as a model for studying human development and disease. The fly genome, 185 million nucleotide pairs comprising just over 13,000 genes, contains counterparts of most human genes, including most of those known to be critical in human diseases. Another widely studied organism, smaller and simpler than Drosophila, is the nematode worm, Caenorhabditis elegans, Figure 138. a harmless relative of the eelworms that attack the roots of crops. This creature develops with clockwork precision from a fertilized egg cell into an adult with exactly 959 body cells, plus a variable number of egg and sperm cells, an unusual degree of regularity for an animal. We now have a minutely detailed description of the sequence of events by which this occurs, as the cells divide, move, and become specialized, according to strict and predictable rules. Its genome, some 97 million nucleotide pairs containing about 19,000 genes, has been sequenced, and a wealth of mutants are available for testing how these genes function. It appears that 70% of human proteins have some counterpart in the worm, and C. elegans, like Drosophila, has proved to be a valuable model for many of the processes that occur in our own bodies. Studies of nematode development, for example, have led to a detailed molecular understanding of programmed cell death, a process by which surplus cells are disposed of in all animals, a topic of great importance for cancer research, discussed in Chapter 18 and 20. Another organism that has provided insights into developmental processes, particularly in vertebrates, is the zebrafish. Figure 139. Because this creature is transparent for the first 2 weeks of its life, it provides an ideal system in which to observe how cells behave during development in a living animal. At the other extreme, mammals are among the most complex of animals, with twice as many genes as Drosophila, 25 times as much DNA per cell, and millions of times more cells in their adult bodies. The mouse has long been used as a model organism in which to study mammalian genetics, development, immunology, and cell biology. New techniques have given it even greater importance. It is now possible to breed mice with deliberately engineered mutations in any specific gene, or with artificially constructed genes introduced into them. In this way, one can test what a given gene is required for and how it functions. And almost every human gene has a counterpart in the mouse, with a similar DNA sequence and function. But humans are not mice, or fish, or worms, or flies, or yeast, and so we also study human beings themselves. Research in many areas of cell biology has been largely driven by medical interests, and a great deal of what we know has come from studies of human cells. The medical database on human cells is enormous, and although naturally occurring mutations in any given gene are rare, the consequences of mutations in thousands of different genes are known without resort to genetic engineering. This is because humans demonstrate the unique behavior of reporting on and recording their own genetic defects. In no other species are billions of individuals so intensively examined, described, and investigated. Nevertheless, the extent of our ignorance is still daunting. The mammalian body is enormously complex, and one might despair of ever understanding how the DNA in a fertilized mouse egg cell makes it generate a mouse, or how the DNA in a human egg cell directs the development of a human. Yet the revelations of molecular biology have made the task seem possible. As much as anything, this new optimism has come from the realization that the genes of one type of animal have close counterparts in most other types of animals, apparently serving similar functions. Figure 140. We all have a common evolutionary origin, and under the surface it seems that we share the same molecular mechanisms. Flies, worms, fish, mice, and humans thus provide a key to understanding how animals in general are made and how their cells operate. Comparing genome sequences reveals life's common heritage. At a molecular level, evolutionary change has been remarkably slow. We can see in present-day organisms many features that have been preserved through more than 3 billion years of life on Earth, or about 1/5 of the age of the universe. This evolutionary conservatism provides the foundation on which the study of molecular biology is built. To set the scene for the chapters that follow, therefore, we end this chapter by considering a little more closely the family relationships and basic similarities among all living things. This topic has been dramatically clarified in the past few years by analysis of genome sequences. The sequences in which the 4 universal nucleotides are strung together to form the DNA of a given species, as discussed in more detail in Chapter 9. DNA sequencing has made it easy to detect family resemblances between genes. If 2 genes from different organisms have closely similar DNA sequences, it is highly probable that both genes descended from a common ancestral gene. Genes and gene products related in this way are said to be homologous. Given the complete genome sequences of representative organisms from all 3 domains of life, archaea, bacteria, and eukaryotes, one can search systematically for homologies that span this enormous evolutionary divide. In this way, we can begin to take stock of the common inheritance of all living things, and to trace life's origins back to the earliest ancestral cells. There are difficulties in this enterprise. Some ancestral genes are lost, and some have changed so much that they are not readily recognizable as relatives. Despite these uncertainties, comparing genome sequences from the most widely separated branches of the tree of life can give us a sense of which genes are fundamental necessities for living cells. A comparison of the complete genomes of 5 bacteria, 1 archaeon, and 1 eukaryote, a yeast, revealed a core set of 239 families of protein-coding genes that have representatives in all 3 domains. Most of these genes can be assigned a function, with the largest number of shared gene families being involved in amino acid metabolism and transport, and in the production and function of ribosomes. Thus, the minimum number of genes needed for a cell to be viable in today's environment is probably not much less than 200 to 300. The most streamlined genome recorded to date is that of a bacterium called Carcinella rudi, which lives inside specialized cells in plant lice and has 182 genes. This organism, however, depends on the genes of its insect host to carry out many of its essential functions. Most organisms possess significantly more than the estimated minimum couple of 100 genes. Even prokaryotes, frugal cells that carry very little superfluous genetic baggage, typically have genomes that contain at least 1 million nucleotide pairs and encode 1,000 to 8,000 genes. With these few thousand genes, bacteria are able to thrive in even the most hostile environments on Earth. The compact genomes of typical bacteria are dwarfed by the genomes of typical eukaryotes. The human genome, for example, contains about 700 times more DNA than the E. coli genome, and the genome of a fern contains about 100 times more than that of a human. Figure 141. In terms of gene numbers, however, the differences are not so great. We have only about 7 times as many genes as E. coli if we count a gene as a stretch of DNA that contains the specifications for a protein molecule. Moreover, many of our 24,000 protein-coding genes and corresponding proteins themselves fall into closely related family groups, such as the family of hemoglobins, which has 9 closely related members in humans. The number of fundamentally different proteins in a human is thus not very many times more than in a bacterium, and the number of human genes that have identifiable counterparts in the bacterium is a significant fraction of the total. The rest of our human DNA, the vast bulk that does not code for protein or for functional RNA molecules, is a mixture of sequences that help regulate the expression of the genes, and sequences that seem to be dispensable junk retained like a mass of old papers because if there is no pressure to keep an archive small, it is easier to save everything than to sort out the valuable information and discard the rest. The large quantity of regulatory DNA allows for enormous complexity and sophistication in the way different genes in a eukaryotic multicellular organism are brought into action at different times and places. But the basic list of parts, the set of proteins that our cells can make as specified by the DNA, is not much longer than the parts list of an automobile, and many of those parts are common not only to all animals but also to the entire living world. That a length of DNA can program the growth, development, and reproduction of living cells and complex organisms is truly an amazing phenomenon. In the rest of this book, we will try to explain how cells work by examining their component parts and seeing how these parts work together, and by investigating how the genome of each cell directs the manufacture of the components so as to reproduce and run each living thing. Essential concepts. Cells are the fundamental units of life. All present-day cells are believed to have evolved from an ancestral cell that existed more than 3 billion years ago. All cells, and hence all living things, grow, convert energy from one form to another, sense and respond to their environment, and reproduce themselves. All cells are enclosed by a plasma membrane that separates the inside of the cell from the environment. All cells contain DNA as a store of genetic information and use it to guide the synthesis of RNA molecules and of proteins. Cells in a multicellular organism, though they all contain the same DNA, can be very different. They turn on different sets of genes according to their developmental history and to cues they receive from their environment. Cells of animal and plant tissues are typically 5 to 20 micrometers in diameter and can be seen with a light microscope, which also reveals some of their internal components or organelles. The electron microscope permits the smaller organelles and even individual large molecules to be seen, but specimens require elaborate preparation and cannot be viewed alive. The simplest of present-day living cells are prokaryotes. Although they contain DNA, they lack a nucleus and other organelles and probably resemble most closely the ancestral cell. Different species of prokaryotes are diverse in their chemical capabilities and inhabit an amazingly wide range of habitats. Two fundamental evolutionary subdivisions are recognized: bacteria and archaea. Eukaryotic cells possess a nucleus and other organelles not found in prokaryotes. They probably evolved in a series of stages. An important step appears to have been the acquisition of mitochondria, which are thought to have originated from bacteria engulfed by an ancestral eukaryotic cell. The nucleus is the most prominent organelle in most plant and animal cells. It contains the genetic information of the organism stored in DNA molecules. The rest of the cell's contents, apart from the nucleus, constitute the cytoplasm. The cytoplasm includes all of the cell's contents outside the nucleus. It contains a variety of membrane-enclosed organelles with specialized chemical functions. Mitochondria carry out the oxidation of food molecules. In plant cells, chloroplasts perform photosynthesis. The endoplasmic reticulum, the Golgi apparatus, and lysosomes permit cells to synthesize complex molecules for export from the cell and for insertion in cell membranes, and to import and digest large molecules. Outside the membrane-enclosed organelles in the cytoplasm is the cytosol, a concentrated mixture of large and small molecules that carry out many essential biochemical processes. The cytoskeleton extends throughout the cytoplasm. This system of protein filaments is responsible for cell shape and movement and for the transport of organelles and molecules from one location to another in the cytoplasm. Free-living, single-celled eukaryotic microorganisms include some of the most complex eukaryotic cells known, and they are able to swim, mate, hunt, and devour food. An animal, plant, or fungus consists of diverse eukaryotic cell types, all derived from a single fertilized egg cell. The number of such cells cooperating to form a large multicellular organism, such as a human, runs into thousands of billions. Biologists have chosen a small number of model organisms to study closely. These include the bacterium E. coli, brewer's yeast, a nematode worm, a fly, a small plant, a fish, a mouse, and the human species itself. Although the minimum number of genes needed for a viable cell is less than 400, most cells contain significantly more. Yet even such a complex organism as a human has only about 24,000 protein-coding genes, twice as many as a fly, and 7 times as many as E. coli.